lecture, and then on bins and bold, the barcode database. Then we'll do about 1 hour working with the software ontology, where I'll just sort of walk you all through the software. Then we'll do some, some more work in R, okay? We'll work a little bit more on tidyverse data analysis. Then we'll have lunch, and then the afternoon, the whole time will be in the lab, okay? And so a little bit less going back and forth today. Good? Okay. So we'll talk about bold barcode of life database this morning. So we'll tell you about what bold is, overview, what bins are, and or barcode index numbers, and how the best features of bold, how we use it, and future plans. Okay, who here has used bold? Who's been on the website? Anybody? One? No? Okay, those? Okay, cool. So the website is boldsystems.org. You can go there if you want during this lecture, no problem. So it's the database for the whole world DNA barcode data. It also allows you to work with the barcode data online on a workbench. You can use it to identify species with the DNA barcode sequences. And it provides reference libraries that we use to identify species. Okay? Bold is structured around individual specimen records. Specimens have a sample ID, which the person who collects the specimen provides. So for example, Ximena's data, you will give us the sample ID that's related to your project. And then a process ID, which is on bold. What is this? Each record has to have at least one sequence, but can have more, because you can sequence multiple markers for each species, or each specimen. Records have projects, so we will download records for some of our projects together as a class. But you can take records from a project and put them into different data sets. And on bold, the sequence gets a taxonomic identification. And there's a lot of taxonomists and taxonomic identification systems that work on bold. Okay? Any questions so far? We will do some examples today in the lecture showing you how bold works. When you upload data to bold, you go on the workbench, which is sort of you sort of log in to the workbench and you can upload data. You can keep your data private until you publish your paper. So say you have new species, you can keep it all just to you. And then when your paper is published, maybe a manuscript describing new species, then you can publish the barcodes. And then you make your data public, and anybody can download it. And so for this course, the data that Robin and I worked with when we were at Los Amigos in Madre de Dios, you will download that data for the course that we made public for this course. And you also can put sequences on GenBank, which is sort of a bigger non-barcode specific sequence repository. So, records on bold specimens look like this. You can see here, this page. Records have a photograph of the specimen picture, which is, you can see here. Coordinates, location, so latitude, longitude, and taxonomic identification. So let's see where we can see here. So here you can see, it's a little bit small, but this is identified to family Vespidae, so it's like it's a wasp, but we don't have any information about genus. There's a subfamily as well. And having all this information, photos and everything, makes this data highly valuable and better than typical data that's on GenBank. So this is sort of the unique feature of bold, is that we have all this metadata for all the records, so it's higher quality data for reference. On bold, many times we are dealing with specimens or species that have not been identified or have not been described by taxonomists. Okay, so they're new species, and so what we do is we assign different species that have similar or different genetic groups that have similar barcodes, what's called barcode index number, or bin. So you'll hear the word bin a lot in this course. And these are our sort of operational taxonomic units. So we treat these as similar to species when we don't know what the species are. Actually, the data we'll work with will have a species very similar to this one, this bin. And what we do is we use a clustering algorithm. It's called RESL, R-E-S-L, and that's what we use to sort of say, okay, these barcodes are part of this bin, these barcodes are part of that bin, and I'll go into that in a little bit more detail. And the goal in the long term is to associate bins with the formal Linnaean taxonomic system. So we sort of have every species that's been described in the Linnaean system can match at least a bin. That's the hope in the long term. And so that's why at our organization, the Biodiversity Genomics Center, we employ many taxonomists. I would say probably probably the second most in Canada. I would say Robin, after probably the CNC. Probably. So we hire taxonomists, and they describe the new species that we find or match names of existing species to bins. And this is a very objective way and persistent way that describes species. We create bins using this algorithm. We only work with sequences that are at least 500 bases, which is most of the barcode region. Only Co1 or COI, as some of you say, where the sequences are very well known, less than 1%, so less than 5, are unknown. Sometimes if you sequence something, you can't tell the letters apart sometimes, A's, T's, C's, and G's. So we want very complete sequences. No stop codons or frame shift, meaning it's probably not with the numite or, what was the other thing you used, Robin? Not numite, pseudogenes. Yeah, so we don't want to work on pseudogenes. And we align the sequence, make clusters. Buenos dias, Alex. And we give them a name in a standard format. So bold, letter, 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 nombre, 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 nombre. Who can guess? So that bold AA001, 0001, that's the first bin. Who can guess what species it is? Hint. I'll give you a hint. Any guesses? One person guess. What species? You all know it very well.
One.
Hint. Okay, hint. There's at least one in this room of that species. Human. Yeah. So Naomi got it. So human is the first one. Okay. Okay. So bins, there are sort of molecular taxonomic units. So that's what we use. Often these match to species. So you have one species, one bin, which is what you see here. The blue, one species, one bin, most of the time. Sometimes you'll have one species that actually hits two bins, and that can happen maybe if you have a cryptic species, for example. You have two species that look similar, so people gave them the same name, but in reality there's reproductive isolation, no interbreeding, then you get two bins. Sometimes you might have two species that actually are one bin, and that can be maybe you have a bird, you have a red bird and a yellow bird. People say those are different species, but actually it's just one gene that changes the color, right? So one species. Or you can have more complex situations where you have one bin that matches two species and one of those is shared with another species. You can have some complexity, but biology is complex, right? So it's just how it happens. So that's sort of how we work. Yeah, bins sort of are a good place to start for taxonomists. Okay, so say if you work on a genus, you barcode the genus or in a new area, that gives you a place to start to say, okay, I think this is one species, I think that is one species, to start working. And you can use these taxonomic hypotheses from bins to start designing your research, right? To try to understand a group of organisms. You can sort of see, so here's a figure from Canada where we show how many species are known in blue, okay, in dark blue, and then the number of species we think exist in the Linnaean system. And then in red is how many bins there are. So in Canada, we've got over around 30,000 bins for Diptera, which is a lot more than formally described species. But for some groups, it's a little bit different. So for example, maybe moths are pretty similar, beetles more described than in bins. So it just depends on how much work you do. But we're pretty close in Canada. Sometimes you can find what's called the discordance in the bin by looking at the taxonomy. So what we can see here, sorry it's a little small. This bin, bold, ABZ, or Z, 0652. All of the specimens have the same phylum, arthropods. All of the specimens are insects, Hymenoptera, Elliptidae, but many have one genus and two have another genus, okay? So there's a conflict. So you have 500 with one genus, two with another genus. Probably those two have been identified incorrectly, is what you could say, right? So maybe our taxonomists looked at that and gave it the wrong ID, or it could be real biological variation. Those are two different species that have the same thing. Or contamination as well. The bin system is sensitive, like any system of data, to the data you put in. If you don't have good quality sequences, or you don't have good taxonomic coverage in your sequences, then the bins will have limited information. You can have some cases where bins don't match exactly onto species. The process of speciation can work differently in some groups than in others. So sometimes the bin system works better, sometimes it works less well. It only works right now, the bin system with CO1, so it only works on animals, so plants don't have bins. So maybe people who work on plants can develop bins for plants. And when a new bin is established, it can change a lot because new data can sort of cause it to join another one or disappear, for example. So bins can be a bit unstable until maybe 1 or 2 years after they are made. But like I said before, biology is complex, so you'll never have a system that doesn't reflect sometimes the complexity of biodiversity. So bins are unique compared to other systems of identification of unknown species. Sometimes, for most people, they will define amplicon sequence variants or OTUs, molecular OTUs, and those will be unique to one study at a time. So one scientist will have one name for something, another scientist will have a different name for the same thing. This can happen sometimes in bacterial studies, for example. Whereas bins connect different studies. So if you find a bin and somebody else finds the same thing, it has the same name. So it's a little bit of a nice system to formalize the taxonomy. Bins can also have DOIs, or digital object identifiers, meaning they're stable and you can cite the single bin. So if you want to tell somebody, you can give them a permanent link to go to the website for the bin. Bins are very clearly defined what they are and consistent across studies. Okay, so now we'll talk a little bit more about how all these ideas are integrated onto BOLD with the BOLD portal, data portal. the barcode ID engine. We'll talk a little bit about the workbench, but we won't use it just yet, but we will later. Taxonomy browser, and then we'll talk in the workshops, we'll do the computer workshops about API and Bold Connector, the R package, which I hope we can download on Windows. It should work. I'm going to try this afternoon. Okay, so the Bold data portal, what this allows you to do is build a summary of, it builds a summary of barcode records from something you want. So we have an example. We'll just open here. So here is the Bold page for Universidad Nacional Mayor de San Marcos Museo de Historia Natural. So the Natural History Museum with this university. So you can see on the Bold page, let me make this, see if I can make it a bit bigger. Where's the plus? Where's plus here? So there have been 3,206 specimens from the museum at this institution that have been submitted to Bold. This week I'm going to try to meet Gerardo Ramos, to see if we can make that 10,000, if he lets us. So you can sort of see many of these records have bins, good quality sequences, and there's been 2,300 records with bins here, 1,000 unique species, so very diverse, 1,000 unique bins, I should say. And of those bins, 893 have species names. So these have been found in 3 countries, so meaning the specimens are from 3 countries, so Peru and probably a couple others, we can see a map, but only one institution. You can see here some photos, so mostly, butterflies. Pretty nice. So you can see here. So you can see here if you click, for example, taxon, the bin name, so here, who identified it, who collected it, and country ocean, so Juan Dorados in 2017. So that's that one. You can keep going. Let's see. Look at that. You guys have some very amazing species here. So it's also Juan Dorados. See? I'll show, hopefully this works. So we can do a little more. So sequencing centers. So you can see most of the sequences for this institution were actually done at the Center for Biodiversity Genomics in Guelph, so where we work. So probably there were samples that were collected and shipped up to us, we sequence, and then we either keep them there or we send them back. Some were done at the Chinese Academy of Sciences South China Botanic Garden, so that's probably plants, I imagine. And some were sequenced here at the university, so that's pretty cool. Maybe Jorge did that. Yeah. You can see here's a little small. See if I can. I wonder if I can make this bigger. Does this work? So you can see here. This is the collection site. So you can see the bigger dots mean more specimens. So mostly from Peru, one from Bolivia, and then one from what's that country? Is that Costa Rica? That one?
Ecuador.
Ecuador? No,
Colombia.
what's this one?
Colombia.
No, this one.
Ah, Costa Rica.
Costa Rica? Sí. I think none are in it because that's Ecuador. I don't think there's any in Ecuador. I think there's only 3 countries, right? So none in Ecuador, none in Colombia, one Bolivia. Yeah. Okay, so here's actually where you can see, so if you wanted to say, where are the specimens held? All of them are here. So you can see that. So actually, so probably what, maybe just a bit of DNA or a bit of tissue was mailed to the University of Guelph, and then we kept that, but the specimens are all here. So if you wanted to find those butterflies in the picture that he posts us, you can just go to the museum and talk to an artist. Okay? And you can see here what the barcodes are. So it's actually a lot of birds, insects. I don't know how you go over. Zoom out. Plants, right? So some people work on plants. Mammals, right? So pretty cool. And then you can see the individual records here. It just shows up to 1,000, but if you want, you can see the actual record. So here's a fish. Let's see. See if it works. That's the specimen page. So this is sequence page. Is there just no picture on anything? Or back to the specimen page?
The specimen.
What is that? I don't think there is one. Ah, it's Jorge. So Jorge collected that fish. Let's try this one. Data sets. I don't know what the specimen is.
Process ID that's generally that sequence.
Yeah.
The
I just don't see it.
specimen.
That's okay. But what you can do if you want, say, to see the specimen. So this is actually one that Jorge collected in northern Peru. You can see here, that's the actual barcode sequence right here. Okay, so that right there is the series of A's, T, C's, and G's that Jorge found for that specimen's barcode. If you want to search a different one, for example, you can just find a species that you want to go to. So say you have a bin that you studied, you want to find, you just type old, I hope it works, you've been having, I didn't do a colon, there it is. Old ADT 0, 2, 1, 0. It's gonna work. You've been having some computer, well, normally bold works pretty well, I think bold is sleeping now. Oh, there it is, okay, let's see this one. So if you wanted to find a specific bin, you just go on the data portal on bold. Portal.boldsystems.com. You can search, and then you can learn about the bin. So look at that. Nice pentatomid, I believe. Stink bug. Yep. So you can see there. Okay, any questions about Bold Data Portal so far?
Teacher,
Yeah?
what's the difference in NCBI database and board system?
Questions. What's the difference between NCBI database and Bold system? So, it's a really good question. There's a lot of similarities in that there's information about the sequence and the gene, but the barcode database, or BOLD, aims to be a much more specialized and data-rich database for sequences. So NCBI, you don't need photos, you don't need physical specimens. Often the records are not curated with a taxonomic system. They don't get bins, for example. So the barcode database is sort of like NCBI with more information that we want for biodiversity study. Yeah. Anything you want to add there, Robin?
No, just NCBI is the sort of very generic, it's the public database for all DNA sequences, for genomics, for everything. So that's like the all-purpose thing. BOLD is much more specialized for barcoding, but it has much more features that are useful for what we want to do with barcoding, the photographs, the specimen, the metadata. I think it's much better than GenBank for all the attached information.
So all data from BOLD can go to GenBank, but not all data from GenBank can go to BOLD. Does that make sense? Right? Because most of the data on GenBank is not rich enough. Okay. So later we'll spend some time on the BOLD workbench, not today. But if you want to work with your own barcode data, you work on the workbench where you can sort of see how many specimens you have, you can manage specimens, you edit information about the specimens, that's where you upload data. You can make custom data sets, you can search by different records, search by geography or bin. So this is when you really want to sort of do work on BOLD. you work there. The workbench is, I think, version 4 now, but version 5 should come soon. The workbench also, the workbench you can make trees as well, like here. So if you want to visualize how your different bins are related, they make a phylogeny or neighbor joining tree. And Bold also has the ID engine, right, which is what you use to identify sequences one at a time, or many at once. And also is associated with a few other things like the R package, Bold Connect R, and a database that we, Robin, myself, and our colleague Sean recently made, which is called Bold Distilled, which will be published soon. So now we will try using the barcode ID engine. Okay? Okay. So everybody, I'm going to try to make them do a little sequence or something. Everybody watch me, we'll do one, and then we'll have you try one yourself, okay? So I have here 2 sequences. And so when you go to boltsystems.org, you go to barcode ID here. This is where you can identify what's on those sequences. So if you want to use, you pick your reference library. So animal library, public, private, do this one. Species search, genus and species search, or if you want to search very deep, then you can do this. So we'll do this, why not? And then you can paste your sequences. Okay, so you paste them in FASTA format. FASTA format is very common, we'll use this a lot. You have the name of a record that is after one of these symbols here, and then the sequence, okay? And same here, name of the record, and then the sequence. And then you push identify. And so it'll run for a minute. We'll come back to this and look. What I want everybody to do now is go to boltsystems.org and go to portal home. Hopefully it works. If it's too slow, we'll quit. But this is thinking. So you can see here, it's thinking. I think maybe the portal's going to be too slow. Let's see again. Okay, let's try this. So bold data portal. So everyone, click that. Bold data portal. We might be having some computer problems back at the University of Guelph, so we'll see. Okay. Everyone's here? Bold data portal. You go launch portal. And then you can search. Let's see if this one works. So you type bold colon keyboard script here. I don't think that is fine. Anything. See if this works. Should fill. Bold AA I. It's a little slow. Let's see. 6, 8. I think it's too slow. Is anybody seeing anything when you take this? Is anything popping up or not? Up in the detail? I think Bold's having a bad day, everyone. I'm sorry. Bold is sad today. It's loading, yeah. Oh yeah, okay, so yeah, you can click one and then just go to search. For me, Bold's having a bad day. This is still going. This is crazy. It's gonna be rapid. that?
Because they're supposed to be bold
Yeah.
and it's coming over.
It's just been like bubbling. Okay, so for some of you, bold might work. So if you just type something like AAI, you can just click. Oh, it went away. Uh-oh. I don't know why that happened. Let's try again. Okay, maybe bold is just too sad today, so lots of people using it. Okay, well, we'll come back to this later. We'll skip this thing that I had planned because bold is not working. I'm gonna tell Paul that it's not doing good. We'll see. We'll try later if bold is better, but I think it's some issues today. But what you could do if bold was working is you could search a sequence, find a species, find a barcode, and identify it. Okay?
Yeah, there was bold days and it's Friday to Saturday, but
Yeah,
it should be
should be
next
fine.
month.
I have to let home. It's not really great, but it doesn't work. So yeah, some people have used bold to identify cryptic species. So for example, around where is this area? Is this part of Peru? Can't remember where this was. But this research team studied a single species, and what they found were there was clear different clusters, which they're showing in these different colors here on the tree. So these ones in pink are all basically the same here, and these ones are all very similar. These ones are all very similar. So each of these groups represents probably a different species. And so this one species that this group was studying actually turned out to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 maybe, right? So you can get your barcodes for your group across the landscape, put it on bold, and then bold turns one species into 13 bins. And so that's probably similar to 13 species. So yeah, you can get data from bold in 3 ways. One is bold releases data packages that you can download. So you just go to bold data and then data spreadsheets, right? Right here. Here, data spreadsheets. You can go in the workbench, or you can use R directly, bold connect R, which is a package that our colleague Samir developed. And you can access bold data from R, which is what we will do in the course. So whenever I download data, and I don't know about you, Robin, but whenever I download data now, I always use bold connect R. I don't ever do it from bold. It's just the best way. It's fast. Anytime the website's being slow on bold connect R works. Let me see if this is going faster yet. Okay, fine. Okay. So this finished. So we'll just go back. So I searched the sequences with the barcode ID. So I had 2 names of my file. I had PHAUS 5241.25 and 1308.25. So that's the first sequence. Or sorry, the second sequence and the first one. So I changed the order. Here. And the first one identified to species, Cephanta acuta, and the second one, it only identified it to family, right? So the first one was a Hemiptera, a bug. Second one, a butterfly, Lepidoptera. And the confidence in the level of ID that it has is 100%. So based on the barcode, we're very confident the first one is this species, and we're very confident that second one is that family. And then if you want to look, you can see the different records that match this. So you can open the different ones here. Off of page US. Just going slow. So yeah, so you can see the different records that were used to help you identify your species. This is still going really slow, so I'll skip this. Okay. Now, just briefly going forward, bold will change in a few ways. Instead of hosting bold only at servers at the University of Guelph, which, as you can see, are quite slow sometimes. Maybe people in Europe are working a lot, and it's making it hard for us to work. We're increasingly going to allow other countries to host parts of Bold. So, for example, if somebody here wanted to host Bold on their own servers, a mirror, then they can do it in Peru. So that way, we have more computer support for researchers using Bold. As barcoding has got more popular, more people use Bold, the computers that we use to host Bold haven't really changed in the last 10 years, actually. They're still old computers, so that way it can be quite slow. So there's a mirror now, I think, that's being hosted in the Netherlands or pretty soon, right, Robin?
Yeah.
Naturalis. Very soon, like we'll do today and this week, we're going to allow people to upload data to Bold in an easier way using our software, which is Ontology here, running on Mac. And that means you don't have to go to the workbench to upload your data. You just go right from your computer. It really is a lot simpler. And yeah, there's going to be more data releases from Bold and new custom reference libraries that we've made called Bold Distilled. Some other things people are worrying about or wondering about is maybe how we can use bins to automate species descriptions. So if any of you have ever worked with taxonomists or you are a taxonomist, you know it's very slow to describe new species. But without taxonomic attention, those species don't get names. So this question's going now about whether you can quickly describe a couple hundred new species based on bins, or whether you still need to use expert taxonomy. And so right now, if we describe only 18,000 species per year, it will take at least 400 years to describe the rest with the current effort, which is a very long time. And so if we want to do it, say, in the next 100 years or 50 years, we need to have different strategies. Okay, so just to wrap up this lecture, Bold is the very metadata-rich repository for barcode data. So it's like NCBI, but it has a lot more metadata, meaning information about each record. Bins gives you bins. Bins are stable, reproducible species proxies, and they allow you to do research at the species level without species names. And really, Bold and bins and knowing how to use these are really the foundational tools and concepts for biodiversity genomics. So even if you study frogs or fish, groups that are well known, many times you can use bins to find new species, for example, or birds. Okay, any questions about Bold bins before we move on to ontology? Yeah, honey?
Confidence in what is the similarity of the two chains?
So what the question is, what does the confidence that Bold gives you mean? What it means is whether or not, so it's less confident when there are multiple different, so say, let's say that your barcode is equally similar to two genera, then that's low confidence, I would say. So confidence would say all the records are in agreement that it's the same thing. Yeah, Robin, do you want to have a step?
It's related to sequence identity, but it's not exactly the same as sequence identity. And I don't know what the precise calculation is that it does. But I mean, suppose you have, it may be that you have an exact match of your sequence to an exact species, and there are no other species that have that exact sequence. Then it would give you 100% confidence. But if there's 100% match to, there may be more than one species with the same sequence, it would give you a slightly lower probability that it's that species, if that makes sense.
Mm-hmm. We can explore this in the course as well. Yeah.
And then maybe it would give you 100% confidence to a higher level, like a genus or a family. Like maybe it doesn't have any exact matches, but it has a lot of close matches which are all in the same genus or all in the same family. So that's where it would give whichever it has the most confidence.
Any other questions about being Bold? Yeah, sure.
About that, what is the similarity threshold is to typically used to make make a valid match, no?
Yeah, what's the question is, what is the percent similarity of a sequence to an existing record that is used to make a valid match? Typically, based on some research that people have done in the past, the amount of variation you observe in most species is about 2%, 2 to 2.2%. So that's what we usually use. If it's like 98.8% or higher, we say that's a match. And if it's less, we say it's not. But it's a bit arbitrary. Yeah. So that's why we like to deal in sometimes more variable confidence. Yeah. Any more questions? Okay. So now I'm going to switch to my Mac computer. So the screen will go off, and I hope you can reconnect quickly. Okay, so, where, slide, play, wrong day. Okay, so now we'll have a short lecture about ontology, which is the software that we made. Then we'll use it together to do barcoding and bioinformatics. And we'll show you how we do it. Later in the course, we have a couple Macs, Mac computers available. So ontology right now only works on Mac because that's what we use. But later in the course, we have, I think maybe between us, 2 more Macs. And so we can give a couple of you them to work on. Okay. So, our graphic designer has designed a logo too, which I think is cool. And the green and the blue are the colors of the Biodiversity Genomics Center, CBG. Okay, so here, I should say lecture 5, we changed the order a bit. We'll talk about what ontology is, why we made it, why we're making it, a bit of history and the plan to make it so that you can all use it, and how to use it. Okay, so why we made ontology is that there are, I didn't tell Robin that I was going to put him on here. Barcoding all life on Earth is very challenging for several reasons. One is that sequencers have traditionally been extremely expensive and only suitable for very, very big core facilities, meaning you can't have them in your lab. Okay? They look like refrigerators. They can have contracts that cost, like a sequel 2 might cost like 300 to 500,000 solas per year just for the contract to use it, right, for a lab. So it's crazy. That's too much money for a lab to use, right? And also, people who are as good at molecular biology like Robin typically are concentrated in high-income countries. So, meaning in countries where there's lots of biodiversity, often there's not as much training in molecular biology. So it's very challenging to have the level of expertise that you need to do very complex workflows without the level of training. Of course, people here are still very skilled, but there's just a bias in the way that resources and investment goes in the world. Next, getting specimens out of many countries to the core facilities, so say specimens from Peru to Canada, is very challenging. This is just the steps required to start applying for a permit to get specimens from Peru to Canada. So it takes a very long time. You have to navigate the bureaucracy of Peru, which is a lot, and it's very hard. So, and last, shipping of laboratory reagents is often historically not viable because you have to keep things very cold, and like the polymerase for PCR, and you can't ship it because it would just die, right? I think this is the only problem now that we can't solve. The only problem we can't solve is that getting specimens out of countries is still difficult. We still can't overcome the bureaucracy of many countries. So, the rest we can solve. For example, with ontology. So I can tell you a bit about why. Because we now have a platform called Ontology, which has software, which is on the computer. That's an old version. We're working on version 0.2 now. And what I call chemware, which is in these plates. We use orange plates now. Robin, do you have those kits here, or did those end up in the lab?
I think they went down to the yeah,
That's okay.
they've seen
So you saw those orange plates that we used yesterday, right, for the PCR? So those have, as Robin explained, freeze-dried chemicals in them. And so basically what we can do now is the freeze drying allows us to ship them at room temperature, right? So we can get them all around the world at room temperature. You can sequence on ONT small sequencers, which cost $1,000, which most people can afford, or it's cheap enough that a couple labs can come together and buy one together. So very cheap to run. And Robin and other people in our lab who have a freeze dryer and all the expertise and all the primers that I can't even make, he can freeze dry the complex chemistry and mail it, so makes it so easy so people in different countries can just add water, right? So it makes it so that you don't need people with PhDs in molecular biology to do barcoding workflows. You can have somebody who you trained in a workshop for 2 days, and then they can do the whole thing, right? So makes it very simple. So what we do with ontology, and this is what you've been doing, right, as a part of this course, you take your DNA from your bug or whatever it is, and water, and you add it to the well. Then, like what you did yesterday, you do the PCR and library preparation. So that's what PCR was yesterday, library preparation today. And then sequencing, which you'll do this afternoon on MinION, which is that small. And that's it. And then you take the software and you go from there. And so we did a test in Peru, Robin and me and Paul. So this is Robin there. So it's Los Amigos Biological Station in Madre de Dios. That's Robin. That's Paul. Hey, I don't know where my arrow is. That's me there in the blue. And we collected about 1,000 insects, which are in these boxes, and some small ones, which are not in the photo. And we'll use those today and show you what we did. But what we found was in about 3 days, we found about 625 species among those 1,000 specimens. About 414 were known, meaning they matched an existing bin with 2.2%, so they were similar to existing bin. And then 208 were new, meaning they were less than 2.2% similar, so more divergent than that from existing bins. So we can do that in 3 days now. And you get data out of ontology that looks very simple, and we'll see this today, where you get your sample IDs, how many reads you had for each of them, and the taxonomy, which is pretty cool. So you can see, without a lot of expertise in bioinformatics or molecular biology, you can do the whole barcoding workflow. And so this is just a bit about the timeline and where we are for some context for the course. So we finished, Robin and the team finished the design, the first iteration of ontology back in September of 2024 when we showed some people in Brazil at a conference. Then we did some testing at CBG around January of this year. As a result of that testing, we spent a lot of time redesigning the user interface of ontology for the last 4 months, and we're just, I think, about to finish that, right, Robin, you'd say? And so this is where we are now. And so what we're working with today is still under development. Okay, so this is a software that's not released to the public yet. We use it internally, but I just say that because there might be some bugs or some glitches that I find that I don't expect. Some things are still unfinished, so you'll see some things look good, some things are still not so good, but that's just what you can expect when we're working with a product that's still under development. We'll do more testing, I think, in September. In November, we will send it to people around the world, some people at Manu Biostation. Is that also a Madrid de Dios? I think it is. Yeah. I think we will make this publicly available probably early next year, I think is a realistic forecast. If you want, you can go to the website, which is www.ontology.bio. And there's a newsletter you can sign up for. We haven't started sending it out yet, but you can get it there if you want updates about when it's available. So I'll write it down. Ontology.bio website.
La página está bloqueada.
So that's there. Okay, so that's the end of the lecture. So let's take 5 minutes, just relax, and then we'll use ontology. We'll do the workflow. So at 10:10, we'll come back, okay? 7 minutes.